And so today I'm going to speak about basically the long-term goal of my lab, which is how can we make our crops better? How can we use basically high-resolution genetic genomic information so we can really develop meaningful strategies to enhance the biology of our crops. So for today's talk, I'm going to focus my talk on one biological process, which is legume nodulation. We'll talk a little bit about that, which is the symbiotic interaction between soil bacteria capable to fix and assimilate for the plant the atmospheric nitrogen and the legume plant itself. And so this long-term goal came basically from the reading of one paper, which was published in 2018 from Ed Buckler from Cornell University. We start basically to develop this kind of time frame of breeding technology in plants. And so at the end of his paper, he left only 3 lines to develop basically the next step, which is what he called breeding 5.0. How can we synthesize new genes, new pathways to improve our crops? And so this is back to 2018. So when I saw this paper, plus also a little bit of frustrations working on biological systems, which are always extremely complex from a cellular perspective, I start to look at maybe the perspective of the implementation of single-cell approaches so we can generate more accurate genetic information. So if we want to generate this kind of de novo genes, module pathways, biological system and traits for our crops, we need to have a very accurate genetic information to know what are the genes we need to work with, in which kind of cells, cell types we need to modify their activity, and what will be the downstream effect of those modifications. If we have these informations, we will be able to implement meaningful strategies. So the talk I'm going to develop today focus mostly on this symbiotic interaction, the nodulation process, and of course this will have impact if we can be successful enhancing the quality of our crops. It will be more sustainable, minimizing the use of nitrogen fertilizer, for instance. But you can imagine the application of such strategies on different kind of biological questions, biological systems. How can we improve our crops' resistance, tolerance to stresses, which could be biotic or abiotic stresses? And so, for instance, in the case of plant pathogen interactions, you can imagine that the single-cell approach would be very valuable to understand how the plants interacting directly with the pathogen react to the presence of the pathogen, how the plant cells which are distant from these interactions are going very likely to develop a systemic response. So trying to understand this different level of response can help developing better strategies to enhance the resistance of crops to pathogens. But then you can imagine similar kind of strategies. It's time to develop new metabolomic pathways, develop new synthesis of metabolites. How can we generate basically new metabolites in crops to increase the quality of our food? And we manipulate those pathways, but we need to understand exactly how they are regulated in which kind of cells if we want to make meaningful decision processes. So this aspect of the research is basically the long-term outcome of single-cell biology. And so one of the limitations is always to acknowledge that a plant is a complex biological system. Cellularly, it's extremely complex. Here, for instance, on this picture, you can see a zoom in with different type of cell layers of the root system, which highlight different cell types composing the root. And each of them has a unique way to use DNA in order to fulfill its biological function. We need to basically, in plants, separate those different cell types to analyze them one by one if we want to understand exactly how the DNA is going to be used differentially between the cell types composing. And so in 2018, 2019, for the first publications, came this idea to decompose the plant into isolated cells, or in the case of my lab, we work with nuclei, and process them using microfluidic systems to analyze their transcriptome one by one. That was basically, in the early days of single-cell biology, the strategy used by laboratories. Now we are moving into, I will say, more complex level of analysis we can conduct at the single-cell level. And so the long-term goal is really to try to capture those programs and then improve them to understand basically how we are going to make our crops better. That will, of course, require the involvement of nanotechnologies, but also synthetic biology strategies. So you have basically 2 schools in the field of single-cell biology in plants, the one which is based on the use of protoplast and the one used based on the use of nuclei. Both biological entities can be used to capture the activity of genes using RNA-seq. With the nuclei, you can also have additional information about the profile of chromatin accessibility, and in the case of the multiome, capturing the 2 informations together at the same time. That's open new possibilities in understanding the regulatory pathways controlling gene expression. When we consider protoplast, of course, you need to access well I mean, you need to access your protoplast, digest the cell wall. Just digestion of the cell walls could be a limitation if you don't have the right cocktail of enzymes, or really, I will say, a unique cell wall composition for specific cell types. One of the limitations also when you generate those protoplasts is the induction of stresses, but also ultimately some bursting, which can create a lot of noise in your data set. So for those reasons, I decided to move right away into the isolation of nuclei to perform basically both gene expression and later chromatin accessibility profile analysis, with the down of RNA leakage, which is basically the loss of the RNA which are located inside the nucleus. So we have done, I will say, quite a lot of work there to minimize those impacts, and I will show you some data later, reporting the fact that now we have hopefully improved into the quality of the information we are collecting from isolated nuclei. So the nuclei has multiple pros. We can isolate them in 20 minutes and process them right away. They are not going to be subject to so much stress responses in this case. We don't need to worry about their size in the microfluidic systems. We can basically analyze the nuclei from any plant species we want. And we have been successful working on wheat, which create basically here one of the biggest challenge, processing those nuclei. We can also, with a multi-omic approach, integrate from those isolated nuclei both gene expression and epigenomic data sets. So you have multiple ways to basically have new information at the single cell. As I told you, the RNA leakage is one of the negative, and it's even worse when you consider that the pool of transcripts in the nucleus is very low compared to what you can collect from the cell. So you need to be very cautious there in the processing of your nuclei. So nevertheless, we have applied our protocol in the lab, Sandra, across different plant species, validated on all the different species, the methodology we have been applying. For today, I'm going to focus my talk on the legume species here for the purpose of studying the modulation process, soybean, common bean, and barrel medic. So in the lab, Sandra optimized step after step the use of those isolated nuclei and their use into a macrofluidic system so we can collect gene expression analysis, and I will show the data today, but also ataxic data sets so we can give another layer of information on our data sets. So when you consider the nodulation process and those three legume species, we have basically three different types of projects running in the lab. The first one is really the early stage of the nodulation process. So here you can see a root of a soybean plant, which is expressing, in this case, a reporter, a promoter specific of the root hair cell, and then the reporter is the TD tomato that we target directly in the nucleus. That gives this very, I would say, unique spotty localization of the red signal. And so why do we focus on the root hair cell? Because this will be the first cell types infected by the bacteria involved in nodulation. So this infection is going to be absolutely critical there because it will lead later to the formation and the infection of the nodule cells. At the same time that this infection occurs, we are going to see also in the cortical and pericyclic cells of the root, the development of the primordia of the node. And here, this is a second project that is supported in the lab by the USDA, collaboration with Senthil Subramanian and Andrew Farmer, where we analyze basically the early developmental stages of the nodule and the lateral root to understand the unique program used by the plant cells to develop a new node. In this case, we are using two different types of reporter, an auxin-inducible promoter upstream to the GFP, but also a cytokinin-inducible promoter upstream to the TD tomato, and that helps us to identify those cells. And then finally, we have the end product of this interaction, which is a new organ, the nodule, on the root of the legume plant, full of this kind of bacteria that you can see in the middle in red, infecting the nodule cells. So this is another project that we are working with, and I'm going to spend some time later today developing this information. So we try to cover basically the entire information, biological processes from the early stage to the later stage of nodulation across three different legume species. So we can perform also comparative genomic analysis and understand better what are the common but also the unique feature of each legume species in interacting with. Oh, thank you. Yeah, okay. So the first project we have here has been developed in collaboration with Florent Frugier and Andrew Farmer, and it was basically the analysis of the infection of the root hair cell of barrel medic or Medicago truncatula by its symbiont. And so we implemented our strategy based on isolation of Medicago truncatula root cells in this case, and used those nuclei to create this UMAP or uniform manifold approximation and projection. This is basically a 2D representation of the transcriptome of all those nuclei, and their relative position reflect basically their transcriptomic signature. And so from this 2D map, you can create basically, in this case, 25 different groups or clusters of nuclei based on their transcriptomic profile. When you generate this information, the first step is really to try to reconciliate 20 years of anatomical observations, more than that in the case of the root, with the molecular profile of the different nuclei we collected. And so for that, we need to functionally validate different type of marker genes in order to functionally annotate those different clusters. And here on this dot plot figure, you can see on the x-axis a list of marker genes that has been described in the literature as specifically expressed in specific cell types of the Medicago root. And here on the y-axis is the profile of the 25 different clusters that we have generated with one Flora. And so those dot plots will reflect two different types of information. The size of the dot here is a reflection of the percentage of the nuclei in the cluster expressing the gene. The larger will be the dot, the higher will be the percentage of nuclei expressing the gene. And the color will reflect the level of activity of the gene in the cluster. So in this case, you can see that multiple genes will have, I will say, pretty well conserved expression pattern in some of the cell types. Here, for instance, in the meristematic cells of the root, or here in the phloem cells of the root. So we can basically reconcile pretty well this type of information, the annotation, the functional annotation of some of those clusters. But by far, we cannot annotate all of them. So what we have done, we perform a comparative genomic analysis with Arabidopsis thaliana root, where we have plenty of those marker genes of the different cell layers composing the root, look for the orthologs in Medicago truncatula and question basically the activity of those orthologs in the UMAP. And you can see a beautiful gradient here of dot plots from epidermal cells to the xylem cells, showing a pretty good conservation of the specific activity of all those orthologs in Medicago across the different cell types composing the root. So it seems here, at least between Arabidopsis thaliana and Medicago truncatula, some of those key genes, very likely programs involved in the differentiation and function of those cell types, are well conserved. And so altogether, you can hand now with the annotation of the 25 different clusters based on their unique cell type specificity. Let's give, I will say, a first, I will say, step in the process of how to use single cell data sets. Now, because we are interested about nodulation process, we can divide our UMAP into two different sub-UMAPs, depending if we treated the root with water or with a symbiont involved in nodulation. You can see that those two UMAPs, of course, look very similar because we have still the same type of cell, the same categorization of cell types involved. But there's one key differences, which is this little red circle. We have one population here in cluster number 2, which is the epidermal cell cluster, which is very unique to the UMAP where we have infected the plants with S. melilot. It does not show up in the mock condition. So when we look at here these small populations, we are going to spend some time analyzing the content of this. But as a first proxy to estimate the quality of the treatment, we look at and explore the activity of genes that people identified during the last 20 years in legumes, Medicago truncatula, and look for the function of those different genes across the 25 different clusters. And so some genes which are critical for the early stage of the infection process has been found again expressed in those epidermal cells, cluster number 1 to 4. But others are more importantly expressed in the cortical cells, endodermal cells, and stem cells. So altogether, in addition to basically confirm the induction or at least the expression of those different cell types during the infection process, we have also more granularity in the information we can collect because now we know exactly in which kind of cell types they are going to be expressed in response to the bacterial infection. So what we have done with Andrew, focusing on, again, this unique population, was to develop what we call transcriptomic trajectories. Basically, you have those two arrows, the yellow one, which does not pass through our unique population of cells, and the green one, which is going to pass through the different population of cells unique to the S. meliloti condition. And so across those two different trajectories, we look at the activity of 4 genes that are absolutely critical to the early stage of the infection of the Medicago root hair cell by the bacteria. And what you can see is this green line will always be characterized by the induction of those 4 genes pretty late across the transcriptomic trajectory, where these inductions will abort very quickly to follow the yellow line. So that's telling us that the co-expression of those 4 genes here at this point in the pseudo timeline is absolutely critical and is a good proxy to estimate the population of cells in the Medicago route infected by the bacteria. So what we have done with Andrew was to question our transcriptomic datasets and to look at what are the genes that show exactly the same pattern of activity, which is basically this long-term induction in the Medicago atlas in those specific cells infected by the symbiont. And we identified a total of 243 genes which show exactly the same profile. 243 genes which could be candidates important for the infection of the router cell by the bacteria. So Sabir in my lab look at the function of those 243 genes and identify here in blue and gray multiple genes associated with biological membranes. This is, I would say, a long-term interest of mine, focusing on the activity of specific genes encoding membrane-associated proteins and how important they are in the infection process of the plant cells by the symbiont. So I will speak a little bit later about some of the work we are doing on other candidate genes here related to the plasma membrane. So this is basically the work we have done so far on the early stage of the infection in Medicago truncatula, and we have a similar work going on the two other species, soybean and common bean. Regarding the work on nodules, which is really the later stage of the infections, we try to provide some context to the work we'll be doing, and that's why for the soybean plants, we developed this single cell resolution atlas, basically applying our single nuclear RNA-seq technology across different soybean organs. It will be seed at different maturation stage, green pod, floral buds, shoot apical meristem, leaves, which could be true leaves or tree foliates, the root, and of course the nodule. This gives us now a collection of 172 different clusters reflecting the cellular complexity of the soybean plant. And so for this analysis, we have over 136,000 nuclei analyzed and identified over 90% of the expressed protein coding genes from the soybean gene. When we look at gene markers specifically expressed in only one cluster among the 172, we were able to identify 124 of those. So that gives basically a first proxy about the information we can collect. We performed some comparative analysis with existing atlases and found that this atlas provided deeper information, in addition, of course, to higher resolution. One analysis we have done recently with Olivier Martin was to try to have a better estimation about the depth of information we can collect from single nuclei RNA-seq. This has been, I would say, a question that people raise for quite a long time. And so what we have done, we estimated for each of the 172 clusters, here each dot is one cluster, we estimated the coverage of the transcriptome based on the saturation of the sequencing. So what we found here is basically that all those different clusters fit pretty well this 1x law that reflects the coverage of the gene expression here on the y-axis versus the number of cells or nuclei in our case analyzed for each cluster. So we identified that if you want a 95% coverage of the transcriptome, we need less than 400 nuclei analyzed. That will give you an information about the quality of the transcriptomic data you can get. You don't need a lot of cells, or even worse, a lot of nuclei to get a precise information about the transcriptome of a specific cell type. Sometimes if you work with very rare cell types, like in this case, you're not going to have, I would say, a good coverage. That means we need to compensate by doing more sequencing or by doing more analysis. But in all cases, most cases, for the atlas we have, we can have over 80% coverage of the transcriptome. So I will say pretty good. Now, another limitation when you develop an atlas like this one, especially in the soybean plant, is the annotation. How do you name each of the 172 cell types composing the atlas? You don't have the same resources like for Arabidopsis thaliana. So for this, we have developed 3 different strategies. The most, I will say, straightforward strategy will be to identify from the literature cell type specific marker genes. Again, the soybean literature is not as extended as the Arabidopsis one. You can identify a few markers, but by far you cannot annotate all the different clusters. As I showed you before, we can also perform comparative transcriptomics, looking at the activity of marker genes from one plant species to another. That could be, I will say, useful. But we have done also an interesting collaboration with a company named Resolve Bioscience and applied their molecular cartography technology. And so in this case, what Setan has done in the lab was to isolate from the root UMAP 4 marker genes for each cluster composing the root of the soybean plant. And so those 4 different marker genes for one cluster will be used to develop specific set of probes and question on a cross-section of the root the activity of those 4 genes. And here you can see the activity of those 4 genes highlighted with yellow dots, red dots, orange dots, and green dots. And so all those 4 genes are all identified on the cross-section as phloem specific. So you can repeat this selection for another cluster for new genes. And in this case, again, you are going to question the cross-section and identify all genes activated specifically in the xylem layer of the cells. So you can continue playing this way and basically adopt a full annotation, in this case of the soybean root, by questioning, using molecular cartography, the activity of selected marker genes for each clusters composing soybean. So the end product is basically this very accurate annotation, which is extremely well supported by functional validation. What was interesting is we also tested this root-specific marker genes selected for molecular cartography against the soybean nodule. And we were able to identify, confirm basically 60% of those marker genes of the root specifically expressed in one cell type of the nodule, which was, I will say, better than I was expecting. What was very interesting is some, of course, marker genes were expressed in a very different cell type. For instance, here you can see in blue and in orange, gene expressed specifically in the inner and outer cortex cells, but also in the vascular, in endodermis. This one is vascular bundle specific of the nodule. And you can see here the activity of those genes for molecular cartography and how they will distribute through the different clusters composing the soybean nodule. As part of a test with this company, we also tried to look for bacterial transcripts, developing specific probes. And here you can see that it's working beautifully. We developed 9 different probes against ribosome diazoefficients, and basically performing hybridizations against our cross-section of the nodule. And here you can see a beautiful hybridization of those 9 different set of probes of the infection zone of the nodule. It's working beautifully. What was very interesting to us was that genes that we found expressed specifically in the phloem of the root of the soybean plant were basically also working very nicely as markers of the infected cells of the nodule. We have a perfect superposition between this set of labeled cells with bacterial transcripts, and in this case, those red cells where we use basically root phloem specific marker genes. So as you can see, those belong to 3 different clusters, F, G, and H. I will go back a little bit later. Basically, this diversity of different infected cells of the soybean using the same marker genes. As another way to appreciate the activity of those marker genes and the accuracy of molecular cartography, you can see those green dots here in one specific population of uninfected cells in the infection zone of the nodule. And you can see that it's really, really accurate. You're not going to see any of those kind of detection of transcripts outside of those. So what we have done altogether is basically develop a comprehensive molecular cartographic picture of the nodule using molecular probes, resolved by molecular cartography technology, and that's helped us now to open different investigations, which will be targeted functional genomic assays, single-cell co-expression networks, or also subcellular analysis of transcript localization using this technology. And I'm going to go through the topics now. The first thing is, how do we understand better the nodule biology? So first, we have this UMAP, which is the end product. And among these different clusters, FGH, those 3 clusters are clearly defined based on molecular cartography as infected cells, plant cells by the bacteria. What we have done was to basically collect this information, all those transcripts, and develop a gene network trying to identify key genes that are important during the nodulation process. And so for those who are expert in nodulation, you will identify in red genes that has been characterized in soybean, but also in other legume species as critical for the relationship between the bacteria and the plant cells. In purple, that's a series of transcription factors that are sometimes, in some cases, important to control the nodulation process as well, like for instance, the NIN or the NAC. And finally, in green, we identify some interesting genes that we need to investigate more, which are under the control of multiple transcription factors, but again, very specifically expressed in those clusters, FG. So that gives, again, a list of interesting candidate genes to explore to try to better understand the interactions on cells and the bacteria. Another work we have done was also to try to investigate the differences existing between the transcriptome of those different clusters. For that, we perform a group FG analysis versus the H cluster. FNG are characterized by multiple gene ontology terms: RNA processing, chromatin modification, vesicle-mediated transport. So that reflects pretty well, basically, also the activity of this CCS50A gene in those two clusters, which is a repressor of mitosis. That's basically a reflection of the endoreduplication occurring very likely in those cluster FNG. In cluster H, you are going to see another representations of nodulation terms, but also organonitrogen compound metabolism and biosynthesis. Basically, that means that here you have active nitrogen fixation and conversion. Again, this is well supported by the active expression of leg hemoglobin gene here in this H cluster. You have very different, I would say, population of infected cells, depending if they are going to move into endoreduplication or move into active nitrogen fixation. And that's basically one of the outcomes we can collect from single cell approaches. We can dissociate all the different population of cells. It also helps us to identify new candidate genes like this gene, FWL3, to perform functional genomic assays. So Prince, former lab members, conducted some functional analysis against FWL3. This gene is homologous to a very well-known macrodomain-associated protein. Well-known, I've been working on this one for a few years, FWL1. This gene, FWL1, is critical to maintain the good infection of the plant cells by the bacteria. If you disrupt its functions, you are going to minimize the infection process. And so one of the work we have done with Prince was to investigate the role of FWL3 as part of those macrodomain fraction of the plasma membrane, which is what we suspect to be the function of FWL3. The macrodomains is basically a subregion of the plasma membrane enriched in specific receptors, but also in sphingolipids. And so as part of the proteins localized in those macrodomain-associated proteins, remerin-1 from Medicago and Lotus, but also flotilins, a series of genes studied by Sharon Long in the 2000s, has been demonstrated to be important to control the nodulation process. So we suspect FWL3 will belong to the same kind of subcellular localization. And so what Prince has done was to first perform a Cas9 disruption of the gene and analyze the phenotype. And if on Cas9 against the genes, you can decrease by around 30% the number of nodules per root. But also at the same time, when you have a nodules, you have here with the cyto X green staining, a decrease of the intensity of the staining of the bacteria, which is basically a reflection of a lower amount of bacteria on Cas9 disruption of FWL3. This is over 60% decrease in this case. So that gives, I would say, basically a biological information about the function of FWL3. When we perform both confocal microscopy images, but also transmission electron microscopy observations to look at the subcellular localization of FWL3, you can see this kind of punchy localization in the plasma membrane, which is also here with those gold particles in the plasma membrane, which is very typical of the microdomain association of FWL3. So that's pretty well into this picture that FWL3, like FWL1, is another important player regulator of the nodulation process. To try to understand better this network of proteins, we have done also some co-immunoprecipitation. So we can find, again, multiple proteins from this pie chart associated with the macrodomain fraction. For instance, we have aquaporins, prohibitin, remerins, and vacuolar ATPase. And Sabir in the lab look at more precisely the activity of some of those interactors and identify, again, many of those interactors of FWL3 specifically expressed in those cluster FGH infected cells of the node. So single cell approach here in this case help us to really narrow down to specific cell types, the most important ones, the activity of FWL3. So what we have done more recently, again, in the context of the atlas, was to look at the transcription factor activity. That's what you have here on the x-axis, transcription factor that we can detect expressed across the atlas. And here on the y-axis, you are going to see all the 172 clusters organized per organ. So you can see here that the activity of the genes, this is a co-expression network in this case, the activity of the genes, I would say, is pretty well spread, but sometimes you are going to find some hotspot of co-expression, like in those red boxes. That's tells us that those transcription factors in those specific organs are going to have, I would say, more likely, are going to be more likely being co-expressed together than in other clusters. So that tells us that those transcriptional regulators very likely has a role in determining organ function. But then we decided to basically change a little bit the graph. And in this case, keeping the same order for the transcription factors, we organize the clusters based on cell type, not anymore organ. Here again, you can find time to time, some co-expression of transcription factors in specific cell types, in this case for different organs mixed together. So it means that altogether, those transcriptional regulators, transcription factors, determine, of course, the organ, but also will have an incidence in determining the cell type of the different organs. So that gives, again, an interesting aspect when we focus on the nodule. Here, for instance, we are going to see a unique set of co-expressed transcription factors of the infected cells of the nodule. That will be an interesting set of transcription factors to work with. So again, we can play with the atlas in multiple ways to really gather, I would say, new type of information for biology and for the nodulation process. I want to finish my talk by moving to a very different field, much more, I will say, microscopic. And that's what we have done, basically working with Resolve Bioscience. Here, I want to mention that those two pictures are taken from the same area. It's exactly the same cell we are looking at. In this case, each blue dot will reflect the transcript from one gene. Those yellow dots will reflect the transcript from a different gene. You can see that the subcellular localization of the transcript from gene 1 versus gene 2 is drastically different in those cells. Moreover, what is very interesting to me is you can see this accumulation of transcripts in those blue dots, which is basically the nucleus of the cells, like if you have a retention of the transcript inside the nucleus. So that gives us now a new way to appreciate basically this imaging capabilities, which is an analysis of the subcellular localization of transcript into the cells and basically provide new, I will say, information about the processing of transcripts, how they are going to be dedicated later to translation. And so again, we can look at now genes, cell 1, same cell here. The first genes, we will see an accumulation of transcripts, which is not outstanding, but pretty significant in the nucleus, where gene 2 will not show any transcript in the site, in the nucleus of the cells. And so what Sutton has done in the lab was basically to look at the localization, the percentage of localization in the nucleus for each different transcript across different cells, thousands of them, for the root and for the nodule. And so he ranked those genes for the root based on the lowest retention from 0, in this case, to over 50% of retention for the root cells, and then apply exactly the same order of the genes, but this time providing the information about the retention rate of the nucleus, of the transcript in the nucleus for the nodule in blue. You can see those kind of peaks of retention here in the nodule, suggesting that you have a pretty strong retention of the transcript in the cells in the nodule, but it's not the case anymore in the root organ. So you are going to find basically a cell type specific, but also organ specific retention rate of the transcript, very likely an important process to control later the translation of genes. So that's basically give another information, new informations about the processing of the transcripts. So I want to finish my talk by a quick summary. So this has been basically 6 areas identified 1 year ago as bottlenecks, and we are basically trying to fill some of those gaps. So the Plant Cell Atlas Consortium developed this figure. And so we have been successful using our nuclei-based technology, applying that across different plant species. I didn't show that today, but we have also the capability to capture multiple information from the same nucleus, gene expression and accessibility. Gene networks is very promising in order to try to really highlight unique genes that could be interesting for further investigation and trying to understand later, hopefully, the quality existing between regulation of those genes together. Comparative genomic analysis will be, I will say, a promising avenue. So we can investigate better the conservation of programs between plant species. I've shown you also today the power of trajectories in order to identify candidate genes, showing similar pattern of activity expression. And this is, I will say, an interesting aspect about not just annotation of cell types, but also understanding RNA biology better, the retention rates in the case of the cells I've been describing to you. And so this is the different people working with us on those projects. I mentioned some of them. Most of the work which I showed today has been done by Alan in the lab, as well as Sandra and Sutton. And we have a long-term collaboration with Andy Farmer, who is helping us on multiple projects. And I want to also mention the role of Syngenta, who provide us funding, significant funding, so we can develop those resources. I will be happy to answer any questions.
So much, Marc, for the great talk. questions?
A fantastic talk. Lots of data always makes me very happy. I've got a couple of questions, but the first one is, when you run your UMAP, what kind of distance metric do you use to build up the embedding?
Well, it's a relative distance because it depends really on your starting point. What do you start with? If you start with an organ where you are going to see a lot of transcriptomic diversity, of course, the distance will be quite different in terms of meaning. So it's really, I will say, a relative distance you are generating. After the distance you create is really, you don't control that so much, analytical point of view. What you can control is the clustering itself, which is basically defining the number of groups of cells you want to generate. You can generate basically as many groups as you want from this type of data.
But if you use a Euclidean metric versus a correlative distance, or if you build your own thing, you can change things, right?
Can at this time there is some kind of, you have different type of computational systems you can use for developing. TSNE will be one and UMAP will be another one, but basically they share the same kind of limitation in developing those kind of distances. I heard recently that you have computational biologists who are moving away from those type of representations to give, I would say, more realistic distance between the activity of the different cells or nuclei they are working with. So that's something we are going to explore, but it has not been well adopted so far in the community.
So could you go to any of your slides that show your UMAP embeddings, because there is a question that I have, and I hope that you can answer it. I'm seeing anything that has a relatively high resolution pictures that I can explain a little bit better of what I'm after. So for instance, if we are looking at, I don't know which one yet. Yeah, so this should be fine. Okay. So for instance, if we, oh, this is perfect. This is perfect. So we have the light blue class, right? We've got the light blue class of dots in that embedding right there. And then you have the dark green one as well. So when I'm looking at these embeddings, the cluster that you have of the light blue, the distance between the top of the light blue and the bottom of the light blue is actually larger than some of the interface between the light blue and the green. Does this mean that there is a gradual change in phenotype or cell that you see in there? And are there maybe subclasses that we haven't looked at?
So the relative localization of the clusters on the UMAP doesn't tell you necessarily the biology behind those cells. So that's something to be very cautious. Many people will try to draw those conclusions that the two clusters closely related together should share some kind of similar information. It's not automatic. That's why before we perform, for instance, this integration of cluster F and G for comparative genomics, we look at very carefully what were the differences from a transcriptomic point of view. And they were very similar in this case. After you can represent those type of clusters, I showed 2D, you can represent a 3D map. In this case, two clusters that you believe very distant in the 2D will start to fall together in the 3D. So I will be very cautious about, trying to interpret those UMAPs from a biological perspective without, I will say, a further deeper look on the transcriptome. That's why you have some kind of limitations on those UMAPs. I think those UMAPs are good for categorizing, grouping together. Based on, I will say, after the issue is you are in control of the number of groups you can generate. I can create 100 different groups if I want to, 100 different clusters. The question is, at one point, is it biologically meaningful? What do I want to do at the end? So you have a lot of controls basically on defining those clusters.
How do you define in the end when you're happy with your hyperparameters?
Again, you put some bias there. We try to basically have a way to correlate our clusters from UMAP with what has been described anatomically. We don't want to create necessarily at this point, multiple subcategorizations. You can create after and focus more on a specific group of cells and try to understand, are you going to find subclusters? You can recluster them if you want to as a subset. that's a possibility to refine a little bit your analysis. But you need to have basically a good rationale to support such so it's really a step-by-step. It's very exploratory. It's very time-consuming because at the beginning, when you have your data, you need to first understand the complexity. And at one point, you need to take a decision, and we try to make the most logically well-sounded decisions based on what we know from an anatomical point of view.
Here. Thank you for your talk. Very beautiful data. So I have a question about the last part in your talk. So you kind of mentioned that the retention of the transcripts within the nuclei, like how much, the proportion that was exported into the cytoplasm, maybe also like the other like outside of the cell would matter a lot for in terms of the translational rates. So I guess that like how are you gonna like study that on a like single cell or single nuclei transcriptomic level? Because if you only isolate the nuclei, that wouldn't allow you to detect the proportion of the transcript that was exported.
It's a very good point. And in some ways, identifying those kind of changes in the allocation of transcript in the nucleus versus the cytosol make me think that the nuclear transcriptome now does not reflect well, of course, not the pool of transcript dedicated to translation, and that was not the point of a nuclear transcript. But now you can also raise a question about, is it a good reflection of an active gene? Maybe those transcripts have been synthesized hours before and written in the nucleus. So you need to be very, I think it just creates now more questions and create more complexity in our transcriptomic information and the interpretation of what is a transcriptome. Before a bulk transcriptome, we can say, oh, it's not going to be cell specific. Of course not, and it's just average that. But now a single cell or a single nucleus transcriptome doesn't give you all the answers either. I think there's a need to go back into really the process of RNA biology. And so for this purpose, what we have done in the lab was to perform also some long-read sequencing on the nuclear transcripts, try to estimate, in this case, differential splicing from cell type to cell type. Are we going to find some, for instance, different maturation processes? So we have some data, we are analyzing those right now, trying to understand basically what could be a causality for retention of a transcript in one cell and not in another.
It's a quick follow-up question. So you kind of also showed the, I guess, pros and cons of like single cell transcriptomics using protoplasts versus the single nuclei. Do you think maybe using kind of contrasting the single cell transcriptome you got from the protoplast versus the single nuclei can give you some of the information maybe like kind of like missing from the transcriptome that you can get from the cytoplasm?
It could be possible. So basically a subtraction of the two informations to identify the pool of cytosolic transcripts. That could be a possibility. Again, now I'm very cautious about that. Are we going to find, for instance, some hotspot of concentration in the cytosol of transcripts, which could be maybe a sign of an active translation? So we're working now on analyzing a little bit more now from the cytosolic point of view or molecular cartography datasets to see if we are going to find always a perfect homogeneous distribution of the transcripts, or are we going to find some hotspots? I would say those kind of single cell data provide information which is, I would say, more accurate than before, but still raise some interesting questions.
Thank you.
Okay. Really, really excellent talk. I think the same direction that she was going. Do you see maybe some correlation of those retained mRNAs with like export factors or like features in the mRNAs that could be involved in that, or you think it's splicing related?
We don't know yet. Basically, I show you descriptive information. We are trying to understand the causality. So our easiest way to investigate the differential processing of the transcript for this long-read sequencing analysis and splicing variants. That's the first hypothesis. Very likely, I don't expect that all the retention of transcripts will be explained through this process. What will be interesting also from my perspective is to look at the distribution of those retention between cell types. We saw it very nicely in the infected cells of the nodule. We know those cells are heavily stressed by the presence of the bacteria. So could it be a way for the plant cell, for instance, to be prepared for the stress, synthesizing the transcript, but not exporting the transcript yet in order to prevent protein biosynthesis? It's not needed. But basically, it's a way for Yeah, almost pre-assuming it's going to happen, and so I'm going to prepare myself to respond to stress. It could be one way. Right now, I think that there's multiple hypotheses of them to